Hello everyone, I am Dr. Anju Yogesh Munde and today we are going to discuss about estimation of the bleeding and clotting time. For the convenience, we are going to divide the practical into the two parts. The first one, estimation of bleeding time. The material required is a sterile needle, spirit, filter paper, stopwatch and cotton. Now, the principle behind the practical. The term bleeding is used to indicate the time required for a small cut to cause bleeding and does not necessarily correspond to coagulation time of blood, for it is dependent on the efficiency of tissue. The average bleeding time is about 1 to 3 minutes but may exceed to 8 minutes in few people. Now the procedure. Clean the fingertip with a spirit by using cotton, allow to dry. Puncture the finger deeply by using the sterile needle. Start the stopwatch. The blood should flow freely without squeezing the finger. After the 30 seconds, collect the drop of the blood at a corner of filter paper. Do not touch the skin with a paper. You will get the blood drop. Repeat the step 3 after every 30 seconds. Then you will get the filter paper with your blood drops. Now, when the bleeding ceases, stop the stopwatch. Note the time and that time is bleeding time. For example, my bleeding time of the blood is 3 minutes. Now the clinical significance. Determination of the bleeding time helps to detect the vascular defect and platelet disorders. Prolonged bleeding time is associated with a thrombocytopenia. In case of the von Willebrand's disease, the bleeding time is high with a normal platelet count. It is caused by a defect combined with a factor 7 deficiency. Now the second part, to estimate the clotting time. The material required is a sterile needle, spirit, filter paper, stopwatch and cotton. Now the principle behind the practical. When the blood vessel is cut, the ruptured platelets and the other cells liberate the thromboplastin. This converts the prothrombin into the thrombin in the presence of the cations like calcium. Fibrinogen in the presence of the thrombin is converts into the fibrin threads. These threads trap the red cells forming the blood clot. Thromboplastin is not found free in the interaction of the platelets. Therefore, the production of the thromboplastin is the limiting factor in the reaction and the determinant of the clotting time in the rectified person. The method used for this practical is a capillary method. The normal range of the clotting time is 4 to 9 minutes. Now the procedure. Sterilize the finger with a spirit swab and allow to dry. Then prick the fingertip by using the needle and start the stopwatch. Wipe off the first blood drop and collect the blood in the capillary up to the 2/3 of its length. Now, after the every 30 seconds, break the capillary about 1 cm to find out whether the fibrin string is formed or not. When the fibrin string appears, stop the stopwatch and note down the time. Fibrin clot formation. There are the two pathways, intrinsic pathway and extrinsic pathway. In case of the intrinsic pathway, those cells or the tissues are involved which are directly in contact with the blood and they are getting damaged. They lead to the formation of the intrinsic pathway. While in case of the extrinsic pathway, when there is an external injury, it leads to the formation of the extrinsic pathway. Result. For example, the observed clotting time was a 6 minutes. Thank you so much for observing the video. Please like, share and subscribe.